Hi, and welcome to Headwaters Science Institute. Tonight, we are excited to share some of our amazing student presenters. This summer, we have had a student research program where the students have spent all summer working on a research project of their choice with a science mentor from Headwaters Science Institute. As many of you know, I'm Meg Seifert, and I am the executive director. Spencer, our program director who ran this program this summer, couldn't be here tonight, but I feel super lucky that I'm getting the chance to help these students present their research, and so that everyone at home can see their amazing work. So first up tonight, we have Perry. Hi, Perry. Welcome.
Hello.
And so, Perry, what were you studying this summer?
So I studied the effects of soil salinity in urban areas on bark beetle infestation in the trees in the Lake Tahoe Basin.
Wow, I cannot wait to hear more. So without further ado, I am going to step out and let Perry go ahead and tell us about his amazing work. For those of you guys who are at home, please go ahead and start asking questions. I'm sure Perry would be super excited to answer a couple questions at the end. So here we go, Perry. I'm gonna.
All right. Here we go. Let me share my screen. Okay. So just real quick before I start this, I want to thank my mentor, Hannah, for help with the experiment design and data processing. So my experiment was the effects of urbanization related soil chemistry changes on bark beetle infestation. Bark beetles pose a threat to the environment in multiple ways. They're responsible for declines in tree populations in forests and kill many of the trees that they successfully infest. This can cause many issues, and especially in California, the increased risk of fire. They're known to the species of Scolytinae and the subfamily Scolytus are known to infest the phloem of a tree and consume it, hence the name bark beetles. Correlation was found between the soil chemistry and infestation rates, but not found with the level of urbanization. So my research question was, how does soil salinity in urban areas affect the incidence of bark beetle infestation in trees? The question is two parts. The first being, how does soil salinity vary with proximity to urban areas? The second part being, how does bark beetle infestation intensity vary with soil salinity? My hypothesis was that areas near urbanization would have a higher incidence rate of infestation due to the race salinity. So my materials and methods. For my materials, I used 250 milliliter resealable glass jars, like mason jars, a digital scale, a knife, a digital total dissolved solid meter for water, a trowel, some gallon-sized resealable bags, 20 micron filter paper, and a funnel. For the survey methodology, at the site that I went to, a tree would be chosen arbitrarily. Once the tree is chosen, the selected tree and every tree within 10 meters are surveyed and assigned a condition. I'll get more to that more later. After all of the trees were surveyed, I would collect a soil sample. A small area somewhere near the first tree would be cleared with a trowel until I found bare soil. And then I collected at least 80 grams of soil into the gallon-sized reusable plastic bag. For the soil analysis, 80 grams of soil were added to a container for mixing. 250 grams of distilled water were added to it. I mixed it with a wooden skewer and then poured the entire solution into a funnel with a piece of 20 micron filter paper on it and ran it through into another jar. When it was collected into the jar, I would run it, then clear out the filter and run it through a new clean filter one more time, and then test the solution with a total dissolved solid meter for water. And then I would put the samples in the sealed jars, seal them, and label them with a Sharpie, and take the numbers and put them down in a Excel document. So explanation about the condition values that I made. So each tree was assigned a value from 1 to 6 based on the amount of damage caused by beetle infestation. Classifications are as follows. Number 1 was unaffected. The tree is healthy and shows no signs of current or past infestation. Number 2 is repelled. The tree shows signs of infestation, but all of the pitch tubes are white, yellow, or clear. This is a sign that the tree managed to repel the infestation and it wasn't successful. Number 3 was infested, light. Tree shows red or deep orange pitch tubes, but is otherwise externally healthy. Number 4, infested, medium. Tree shows extensive bark damage and galleries are visible in any exposed lower branch wood. The tree may look dry, but it is not dying. Number 5, infested, heavy. The tree shows extensive damage to all parts and the needles are turning brown and drying up. Number 6 was killed, beetle. The tree has been killed and shows extensive damage from beetles underneath the bark and outside. Null, or killed N/A, not applicable, is used to document artificially felled tree stumps or those that are decomposed beyond diagnosis of mortality. So for my data figures, this chart shows the correlation between the electrical conductivity of the soil and the condition of the trees. It's a bubble graph, so each point, the larger they are, more trees were assigned that value at the site. This is the second chart I made. It shows the values, the same exact values as last graph, but I removed the major outliers. And it shows a lot more correlation, 0.7 compared to 0.2. The outliers were 3 small dead trees that were most likely killed. It showed extensive signs of beetle damage, but they were most likely killed due to the building of a trail right next to them. The figure shows much higher correlation compared to the last graph, as previously mentioned. The p-value was on the order of around 1 times 10 to the negative 18th. So, discussion. My findings did support part of my hypothesis, that being that raised soil salinity will show an increased amount of infestation rate, but it did not show correlation between urbanization and raised soil salinity. The correlation was strong enough to be statistically significant. Some possible improvements for future research. The urban affected areas that were surveyed may have been thinned, and the sample size was relatively small, both the number of sites and number of trees surveyed. So infection research. Some remaining questions are there mapped geologic or other documented properties that can accurately predict infestation risk? Could a larger and more diverse sample size show different correlation? And could a survey with larger differences in terrain change affect the outcome of the study? And here's my works cited. So thanks for watching.
Great job, Perry. Thank you for that. If anyone has questions at home, feel free to type those in. I'm going to go ahead and get started. I love asking questions. And I was curious how you became interested in soil and bark beetles. What prompted you to think of this project?
Yeah, so it was just like several years ago when we were like really in the middle of a big drought in California. I had heard, I was going camping with my dad, and I asked him why there were so many dead trees. And it sparked up a conversation about the bark beetle epidemic, I guess, in several of the pine forests in California. So that's kind of what inspired me to look more into it.
Okay, and what about the connection with soil? Why did you think salinity might affect bark beetles, or what made you choose that?
There was a lot of correlation that I had in other research papers that I had found a lot with. There was some with pH and nitrogen fertilizers, and also with water levels, drought and stuff. So I just wanted to look into a different aspect of soil chemistry that hadn't really been looked into before.
Yeah, we have a question from home. We'll take one more. So why do you think that there might be higher level of salinity in urban settings? Do you have a sense of this?
I had thought that it would be just due to runoff from potentially agricultural reasons or mining or like landscaping. There was no correlation found, however.
Okay, between urban areas and salinity.
Yeah,
Yeah, I would say the other thing is, and not necessarily here, because in the Tahoe Basin we have to be really careful with what we put on the roads so that they
yeah,
don't. But that's a big part of places that have winter, is that a lot of the things used for the roads have have salinity, or have
yeah.
some salt.
Rock salt.
Yeah. We actually have one more question. I'll take this before we move on to our next speaker. So how many trees did you take samples for?
It was just under 100 in total. I think it was somewhere around like 93 trees,
Wow.
approximately.
How much time did that take you?
It was over the course of about a week and a half, I think, the total. It was spaced out. It wasn't every day, but it was about a week and a half of data collection and processing.
Did you enjoy that part of the project?
Somewhat. I learned a lot.
Nice. Well, thank you, Perry. I am going to let you go. Great job, and we're ready to hear some more. All right. Let's go ahead and add Lara into the stream and pull that out. How are you doing?
Good, how are you?
Great. So thank you for joining us tonight. And can you tell us a little bit about what you were researching?
So I researched the eggshell consumption of Western bluebirds in like the different reproductive stages and broods they can have.
Wow, that sounds awesome. All right. Well, I'm super excited to hear about your research. I'm going to go ahead and pop myself out, and you can pull up your presentation so that we can hear all about bluebird eggs.
So I studied the eggshell consumption in the different reproductive stages and broods of the Western bluebird. Thank you so much to my mentor, Jennifer Hebner, for helping me with this project. She was a great help with the manuscript as well as the statistical analyses, and she was very patient with all of my questions. Calcium is very important to birds for reproductive success. Supplementing calcium to birds can increase egg volume, eggshell thickness, and can induce an earlier start of breeding. Insufficient calcium, which can be caused by a low availability of snail shells or calcium-rich soil, can have adverse effects. Some of these include abandoning the nest, abnormalities in eggshells, and a later start of breeding. Arthropods, such as insects, only amount to 5 to 10% of the needed calcium for egg laying. So birds need external sources to attain sufficient calcium. Songbirds do not store calcium in their eggs prior to egg laying, so only enough calcium is retained for routine metabolic needs. Much research has been done on the benefits of calcium supplementation, and there is a lot of evidence that songbirds will consume supplemented calcium, but there has not been research done on the timing of consumption. Which leads me to my question: during which reproductive stages will eggshells be consumed the most, and how the timing and amount of eggshells consumed differ between the 2 to 3 broods bluebirds have? My independent variable was reproductive stage and brood number. My dependent variable was the amount of eggshells consumed by bluebirds. I hypothesized that bluebirds would consume the most eggshells before and while egg laying. This is because birds will not store excessive calcium, as this adds unnecessary weight that requires additional energy usage. Calcium is also important for strong eggshells. I hypothesized that the most eggshells will be eaten during the second brood. Since fledglings are still begging to be fed, and the timing between first and second brood is short, only around 1 to 2 weeks. These are my materials and methods. I provided 5 grams of sterilized eggshells in a small container on top of the bluebird nest box. I ran a 2-way ANOVA. I first tried a type 1 with interaction, which didn't really work because brood number and reproductive stage didn't have any interaction. I then tried to type 2 without interaction, and this worked, and it can be seen in the table on top. I also ran a Tukey post-hoc test. I had a total of 56 for brood, and I had 43 for brood 1 and 13 for brood 2. Reproductive stages ranged from 40 to 50. This is my graph. The most consumption was seen in nest building and egg laying. Although statistical tests did not show any significance in the different broods, there was a general trend that there was more consumption during the second brood. Greater amounts of eggshells were consumed before and during egg laying, supporting my first hypothesis. Birds do not store calcium, which is important for eggshell development. General trends showed that the consumption during the second brood was greater than the first. Statistical tests showed this difference was near significant, a p-value of 0.09. This did not support my second hypothesis. The sample size of the second brood was significantly smaller than the first brood, so the data may not have been entirely representative. There was less data, and this may have been caused by weather conditions that were unfavorable, for example, rain and heat. Some volunteers also dropped out of the study in the later stages. Many of my questions were already answered through literature reviews for future research. I still have one question though. If calcium supplementation increases the intensity of the pigmentation in the eggshells of western bluebirds, I could use a portable color scanner for an objective identification of color. These are my works cited that I used for my presentation as well as what I've used so far for my manuscript. Thank you very much for listening.
Wow, great job. So you may or may not know, but my very first foray into research was studying eastern bluebirds in North Carolina when I was at Davidson College. And so I really love bluebirds, so I was super excited to hear your talk. I'm curious, how did you first become interested in bluebirds?
I've always liked birds since a very, very young age. I got into bluebird monitoring, and I've been doing it for 2 years. And I found them a great test subject because there are many other volunteers in the bluebird club I participate in, and I've loved bluebirds just because I see them every week and I see them grow up.
Yeah, they're pretty special. And what sparked your interest in the calcium consumption?
I had previously done a few small experiments with the birds in my backyard on calcium, and I find it really interesting because I always love seeing the eggs of the bluebirds, and I wondered when they would exactly eat the calcium they need for eggs.
Nice. Does anyone at home have questions? Feel free to type those in. Yeah, I really actually love birds that breed in nest boxes. So I've also studied things like purple martins, wrens, nuthatches, and my PhD work was on barn owls, which have much bigger nest boxes and are really exciting. Have you studied, or does your group work with any other, cavity nesting birds?
The Southern California Bluebird Club focuses on bluebirds, but there's a lot of other projects, and one of my mentors that has been a mentor to me for a long time, Miss Martin, she has an organization that focuses on saving dead trees for those cavity nesting birds, and that includes like woodpeckers and titmice, and there's also other wildlife that use dead trees, so it is really important to them.
Nice. So we have a question from home. Andy Giordano, he's actually a science teacher and our board president. What is the potential evolutionary value is there in the egg coloration?
I am not really sure if egg coloration provides any benefit. For some birds, like our shorebirds that sometimes make scrape nests, they have spotted eggs to camouflage them in the sand. But for western bluebirds, I am not sure because they are inside cavities and would be only seen by the parents.
Okay. And how about what types of birds eat eggshells? Is it just bluebirds or all birds? Do you know anything about that?
A lot of birds will consume calcium, and there have been studies showing songbirds that will consume um, eggshells as well as other sources of similar types of calcium, like snail shells and oyster shells.
Nice. Yeah, in terms of the coloration in bluebirds, I'm not sure if there's an evolutionary value necessarily, but yeah, I don't know. I guess it's an interesting thing to consider. I think it just comes up because of your interest in maybe looking at pigmentation in the eggs in the future, and I think it'd be good for you to think about what connection might be there. All right. Anything else that you want to share with us before?
No, thank you very much for listening.
Yeah, thank you so much. That was a great presentation. I really enjoyed it.